Welcome to our presentation on extinction, biodiversity loss, and ecological monitoring, prepared for CML 6013, Ecological Science. In this video, we explore how biodiversity is disappearing and what tools we use to monitor and prevent this crisis, with special focus on Malaysia and Southeast Asia. Extinction is the permanent disappearance of a species, while biodiversity loss refers to the ongoing decline in the variety and abundance of life forms. Globally, this issue is accelerating due to factors like climate change, pollution, and habitat destruction. In Malaysia, species such as the Malayan tiger and hornbills face critical threats. Our ecosystems, forests, wetlands, reefs, are losing resilience, and urgent actions are needed to reverse these trends. Bioindicators are living organisms that reflect the health of their surrounding environment. They act as early warning systems that alert us to ecological stress before irreversible damage occurs. These indicators help us monitor air, water, and soil quality, providing a cost-effective and real-time method of environmental surveillance. Bioindicators are organisms or biological responses that can reveal the condition of the environment and the presence of pollutants. These species are sensitive to specific environmental changes and can act as early warning systems for ecological disturbances. Because of their strong interactions with their surroundings, bioindicators can reflect the quality of air, water, soil, or overall ecosystem health. Lichens, which are a symbiotic relationship between fungi and algae, are highly sensitive to atmospheric pollutants, particularly sulfur dioxide, SO2, and nitrogen oxides. In urban and industrial areas, lichen diversity is drastically reduced. Their presence or absence is used to assess air pollution levels over time. A study in Kuala Lumpur showed a decline in lichen species near traffic-heavy areas, indicating degraded air quality. Conti and Cecchetti, 2021. Amphibians, such as frogs and salamanders, are key bioindicators of aquatic ecosystem health. Their permeable skin and dual life cycle, aquatic larvae and terrestrial adults, make them particularly vulnerable to pollutants like pesticides, heavy metals, and changes in pH. In Sabah's Crocker Range, researchers noted a decrease in amphibian diversity in areas near oil palm plantations, linked to runoff and chemical leaching. Das and Haas, 2021. By tracking changes in bioindicator populations, scientists can detect environmental degradation before more resilient species begin to decline. These organisms act as canaries in the coal mine, allowing conservationists to prioritize areas for protection and mitigate stressors before large-scale biodiversity loss occurs. This data feeds into red list assessments and biodiversity early warning systems. Monitoring biodiversity requires reliable and efficient techniques that can be applied across various ecosystems. Two of the most commonly used methods in modern ecological studies are remote sensing and camera traps. These tools allow scientists to observe environmental changes and track wildlife populations in a non-invasive and cost-effective way. In some cases, DNA barcoding is also used to detect species through genetic material, especially when visual identification is difficult. Remote sensing involves using satellites or drones to observe land cover, forest loss, vegetation health, and sea surface temperature changes. Tools like NDVI, Normalized Difference Vegetation Index, help monitor forest health, detect illegal logging, and assess habitat changes over time. Remote sensing has been used in Sarawak to map primary forest loss and predict species habitat contraction using satellite imagery and forest classification algorithms. Bryan et al., 2021. Camera traps are motion-sensitive cameras placed in natural habitats to photograph or video wildlife. They are non-invasive and operate continuously, even in remote areas, capturing data on species presence, behavior, and abundance. In peninsular Malaysia, camera traps help document the movement of Malayan tigers, providing critical population data that supported the development of the National Tiger Action Plan. Mohammed et al., 2020. DNA barcoding uses short genetic markers from tissue, soil, or water samples to identify species. This is useful for monitoring cryptic or endangered species and understanding species diversity in a given area without visual observation. Used in Borneo to detect presence of elusive species like clouded leopards through environmental DNA, EDNA, from water bodies. Let's take a closer look at a local case study on biodiversity monitoring, the coral reefs of Tun Sakran Marine Park in Sabah, Malaysia. Located off the coast of Semporna, this marine park is part of the Coral Triangle, a globally important hotspot for marine biodiversity. The reefs here support hundreds of coral species and over 500 species of reef fish and invertebrates. However, these vibrant ecosystems face serious threats from coral bleaching, overfishing, coastal development, and marine pollution. To assess and protect reef health, scientists from Reef Check Malaysia and Sabah parks have implemented several ecological monitoring methods. One of the key techniques used is quadrant sampling, where 1 meter square frames are placed over coral beds to assess the percentage of live coral versus dead coral or algae. Another method, the line intercept transect, involves laying measuring tape along the reef and recording every coral or substrate that touches the line. In addition, visual fish censuses are conducted to monitor indicator species such as butterflyfish and parrotfish, whose presence reflects coral health and ecosystem balance. These monitoring activities are complemented by water quality testing, which tracks changes in temperature, turbidity, and nutrient levels that may be linked to coral bleaching events. The coral reefs themselves act as bioindicators of marine stress, but other species such as the crown-of-thorns starfish and excessive macroalgae also provide insights into environmental pressure. Findings from the 2015 to 2022 surveys revealed significant bleaching events during El Niño years, with live coral cover dropping by up to 40% in some sites. As a result, conservation authorities implemented targeted responses, including expanding no-take zones, regulating tourism activities, and increasing community awareness. This case study demonstrates the power of combining bioindicators and monitoring techniques to inform science-based marine conservation. It also supports Malaysia's broader goals under the Sustainable Development Goal 14, which aims to conserve life below water. Ultimately, this example from Sabah shows how ecological monitoring can drive real-world action to protect fragile ecosystems. In summary, extinction and biodiversity loss are pressing ecological challenges. Bioindicators and monitoring techniques offer vital tools to detect and prevent irreversible damage. Integrating these tools into policy, education, and land use planning is essential for long-term biodiversity protection. Throughout this project, we used various digital tools, Google Scholar and Science Direct for research, WhatsApp and Google Docs for group communication and file sharing, and CapCut, PowerPoint, and 11 Labs for video production. All of the scientific content and case studies presented in this video are supported by journal articles sourced from platforms such as Google Scholar, Science Direct, and ResearchGate. These references include key studies on bioindicators, ecological monitoring techniques, and biodiversity loss both globally and in Malaysia. Using these reliable sources helped us ensure the accuracy, relevance, and academic integrity of our work.